Hello, everyone. Thank you for joining this installment of Boyce-Thompson Institute's Breaking Ground discussion series. My name is AJ Bushy. I'm in the communications department here at BTI. And for this month's Breaking Ground, we welcome Dr. Georg Jander, a plant molecular biologist here at BTI who specializes in the relationships between plants and herbivores. And today we'll be talking about his research into using insects and viruses to protect crops from agricultural threats. So it's a really fascinating topic. We tend to think of, or I tend to think of insects and viruses as being threats. So we'll see how they can be used to protect crops from the threats. So hi, George, welcome. Before we get going, a few housekeeping announcements for those of you who haven't joined one of these before. It's about an hour long. We try to keep it an hour. The first half hour, George and I will have a discussion, and then the last half hour will be open for Q&A, and George will answer some questions. And everyone out there will be muted, and you can ask questions in the chat, the little chat box. And I've also enabled some closed captions, the live transcript function, which is okay with some of the scientific terms. It misses a little bit. And you can hide them if you don't want to see them. There's a little CC button down at the bottom of Zoom. You just click on that and then hide subtitle. So I know we've all been using Zoom for a while. You're probably familiar. But thank you everyone out there for joining us today. This is going to be a really exciting one. I'm sure of it. So George, first, some of you may remember George. We did a Breaking Ground last January. So you may know him a little bit, but for those who didn't see that one, George, would you mind telling us a little bit about yourself and your research?
Okay, well, I guess I'll share the screen first and introduce my lab, who's responsible for that.
Sounds good.
Yeah, so my name is Georg Jander. I'm a faculty member of the Boyce-Thompson Institute, and as AJ mentioned, my lab focuses on plant-insect interactions, and we have a fairly large group at times. This is last summer, where we had a lot of summer undergraduates. So in the summer, typically, my lab is twice as big as it usually is because of visitors from abroad and undergraduates. And the common theme was really looking at plant-insect interactions, and more specifically, in general, chemical defense in plants against the insects that are feeding on these plants. And we have a lot of different projects going on, but today specifically, I'm going to talk about a project that's being done or has been done by Sung-Ho Chung, who is a former postdoc in my lab, and Madia Begum, another former postdoc. So this project has just been completed, and both of them have gone on to other jobs that they're now working on. And specifically, this is a project that was not only in my lab, but involved 6 other labs at other universities. So it was led here through the Boyce-Thompson Institute. And it involved 2 labs with Alan Miller and Steve Witham at Iowa State University, Claire Castile's lab at Cornell, Dan Boytas at University of Minnesota, and Dinesh Kumar and Bryce Falk at UC Davis. And we were all working together on a single project that was funded by DARPA through a project that they called Insect Allies. And I'll go into more detail about why they're calling this overall program Insect Allies and what we're trying to do. But the first question here really is, why is DARPA interested in agriculture, and what is the military interest in agriculture?
Yeah, that was going to be my first question, George, is what exactly is DARPA and why are they interested?
So DARPA stands for Defense Advanced Research Projects Agency, and they're known for funding research that's sort of on the edge of what's possible. So funding, research on RNA vaccines before they became popular, funding the internet before we were all using the internet. And so they were interested in developing methods for countering agricultural threats using new technologies that would be developed through this Insect Allies program. And the kind of threats that DARPA might be interested in is, one of them is illustrated here, is just sort of sporadic random events. So what's on this graph here is corn yield in the United States over a period of 50 years. The overall trend here is upward. So we've had a more than 2-fold increase in average corn yield in the last 50 to 60 years. It's continuing to go up, which is also good. But we have these periodic spikes, downward spikes in yield, and most of them are weather related. Drought is the chief among these. And so one idea would be, rather than breeding plants that are always drought tolerant, which probably wouldn't be very effective because you would have something like a cactus that's putting all its energy in being drought tolerant, what about engineering plants so that we can turn on drought tolerance only when we need it? Or for instance, if we know that it's going to be a drought in the next 2 weeks, we can turn on a drought resistance gene and make these plants drought tolerant, but only temporarily, so they're not always investing in being drought tolerant. Another thing that DARPA was interested in is these emerging threats. This is an illustration here, actually going the other direction from the US to other countries. This is the case of the fall armyworm. The fall armyworm is a native insect here in North America, a caterpillar, and that was accidentally introduced to Africa in 2016. And over the course of 2 years, spread across the entire continent. So by the beginning of 2016 to the end of 2017, the fall armyworm had spread across the entire African continent. And by 2019, it had reached all the way to China. So new infections can spread incredibly rapidly. The kind of damage induced by the fall armyworm is illustrated here. This is an African cornfield here on the next slide. And so here is a before and after picture, before the arrival of the fall armyworm and after, and clearly there's an incredible amount of damage here. And that's something that you might want to combat by genetically engineering corn plants. Or, if you're a plant breeder, you might be interested in this plant here on the right in the after photo, which clearly appears to be resistant to the fall armyworm, whereas the whole rest of the field is being eaten. And you might ask, this is a native North American insect. Why don't we have this problem with fall armyworms? And that's because most of the commercially grown corn in the United States is currently genetically engineered and specifically has insect resistance genes that makes the corn resistance against moths and caterpillars like the fall armyworm.
Is that the BT corn? Is that
That's the BT corn? Yes, exactly. So the BT corn specifically, or most of it, targets butterflies and moths that might be eating the corn, including the fall armyworm. And so this sort of problem no longer occurs in the US, even without the use of insecticides.
Is there any concern of, with DARPA being involved, is there a concern about the militarization of agriculture since they're like a defense?
Yes, that's obviously a concern, but I don't think it's a new concern. My opinion is that as long as we've had agriculture and military, we've had militarization of agriculture going back to Roman times and probably even before then. If you look at stories from the Cold War, which is shown in my next slide here, this is the Colorado potato beetle here in the picture on the left is a North American insect that is the most serious pest of potatoes. The two posters here on the right are from East Germany from the late 1960s. And what you see here in this poster is a USA CIA plane releasing Colorado potato beetles to destroy Soviet agriculture. And so it was well known in Eastern Europe that this was a military sort of invasion, releasing potato beetles into Eastern Europe to destroy the potato crop. Whether or not that is the case is something that we'll never know, but it's certainly something that would be relatively easy to accomplish as a military goal. This next slide is posters from Czechoslovakia. So here on the left, you see some American beetles preparing to invade a Czech potato field. On the right, you have happy Czech school children heading out into the potato fields with their little bottles here to collect potato beetles and save the crop from the American beetle. So my wife, Lucia, who grew up in Czechoslovakia in the 1970s, was a school child in the 1970s, actually was sent out into potato fields just like this to collect potato beetles and combat the infestation. But she tells me that they were not at all happy about going into the potato field. So they didn't look like the children in this drawing here.
So the beetles were there, though. They did get introduced somehow.
They did get introduced somehow. We don't know whether it's intentional, but certainly the rumor in Eastern Europe was that it was intentional, but it could just as easily have been unintentional. Mm-hmm. So for instance, a more recent incident is the corn rootworm, which is another beetle that infects corn roots. That was introduced more recently in Europe. And the first infestations were actually in Yugoslavia around US military bases during the Bosnian War. And so you could imagine that, if thousands of tons of equipment are brought over there from Midwestern military bases, maybe some of these potato beetles hitched a ride on that equipment. And so both accidental and intentional introductions of this kind are probably happening all the time. And some of them turn out into major outbreaks. And that's one thing that we'd like to be combating. And ideally, not by sending children out into the fields for mechanical control of insects, but having more effective systems.
Right. So that's a good segue into, what are you doing to help protect against these kinds of threats?
Yeah. So anyway, the vision of DARPA is what I've illustrated here in the next slide. They put out a call for proposals to specifically come up with a rapid response team for agricultural threats. And the general idea of what they were asking us to do, or asking applicants to do, is illustrated here in this slide, is that you'd have a source plant that's infected with viruses. An insect picks up this virus and transfers it to a target plant. The virus starts to replicate, and whatever transgene that you've cloned into that virus is now expressed in that plant. And that plant, as I've illustrated here in this case, turns from dark green to light green because that virus encoded gene is expressed. The insect that's been transmitting that virus would die. It's been engineered so it can't reproduce. And meanwhile, the virus has also been engineered so it can't be further transmitted by some other insect that's coming in this field. Specifically, in our case, what we have is a virus that's lacking its coat protein, which I've illustrated here by this yellow hexagon, which is expressed in the source plant, but not in the virus itself. So the virus can replicate in the target plant, but without its coat protein, it can't be picked up again by an insect. And so this is a one-time infection, and we're expressing whatever transgene we want in that plant, and thereby creating some sort of resistance or tolerance phenotype in these plants. So this was sort of the mission statement of DARPA. They have a general idea saying, engineer us an insect vectored virus that can make a plant resistant to something, make sure that the virus can't get retransmitted, and make sure that the insect that's doing the transmission will die after the transmission. And so our team of 7 labs that I showed at the beginning submitted a proposal of this kind, and then had a specific target in mind, actually several targets. I'm going to only talk about one of them. That's illustrated here in this next slide. So within this broad frame of the DARPA call, what we decided to focus on is a specific plant insect virus system. And looking at sugarcane mosaic virus and foxtail mosaic virus, these are 2 well-studied viruses that are known to infect corn. They're both can be transmitted by corn leaf aphids, 2 different corn plants. We did all of our experiments in the greenhouse, of course, so we wouldn't want to infect transgenic viruses out in the field. And then we cloned genes into these viruses that would make the corn plants resistant to fall armyworms. So that was what we proposed in our DARPA proposal, and that was what was accepted. And then I'd like to illustrate a little bit of how this works. Yeah. So one initial issue that we had is that viruses are very small in terms of the DNA or RNA that they contain, and they're not very tolerant of putting in big genes. So AJ, for instance, mentioned the Bt toxin that's making corn resistant to fall armyworms. That is a huge protein, and we wouldn't be able to clone it into viruses. So instead, we looked around in nature and say, what proteins are out there that kill insects that are quite small? And one thing that we came across are spider and scorpion venom toxins. So as you might imagine, spiders and scorpions are very efficient at eating insects. And in their venom, in their salivary venom, they have very small insecticidal proteins that they're injecting into the insects to kill them very quickly. And another class of small proteins that we used are so-called lectins from Pinellia ternata, and also here from the snowdrop plant. So small proteins that bind to the insect gut and sort of destroy the gut membrane. And we chose these in particular because we thought they would have synergistic activities with the insect toxin. So the insect is eating not only the lectin from the snowdrop, but also the toxin from one of these spiders, And that means the spider toxin would get into the insect body more quickly after it's being eaten. So normally when it gets injected by a spider, it goes directly into the blood of the insect. But if it's eaten orally, it's not as effective unless we also have this snowdrop lectin involved. And so we cloned up to 3 of these genes at a time into viruses and then looked to see what happens to insect resistance. So here we have in the next slide, one such experiment. We've done many dozens of such experiments. So here we have fall armyworm caterpillars and growing on 2 different maize plants. On the top, we have caterpillars that have been growing on so-called wild-type maize. And on the bottom, we have caterpillars that were growing on maize that were infected with sugarcane mosaic virus and also NC1A, a protein from Nephila clavata, this orb-weaving spider that's insecticidal. And on average, these caterpillars on the plants that are infected with our transgenic virus are smaller than the plants that were growing on the uninfected maize plants or the maize plants infected with a control virus. And so this was overall successful, and we also saw synergistic effects now. So if we put the spider venom protein and also a plant lectin, or the spider venom and a scorpion venom and a plant lectin, sort of stacking 3 of these different genes, we got progressively better caterpillar resistance in these maize plants. And we were also able to show that this virus now that's carrying the spider venom gene can be transmitted by the aphids. The aphids are transmitting the virus to the target plants, and we've made it so the aphids then are able to transmit the virus, and they're now engineered aphids that die after 2 weeks. In addition, they're also feeding now on these plants that are containing the insecticidal toxin. And so within 2 weeks, we have the virus transmitted, the insects are dead, and we have a virus that's lacking its covering, its code protein, so it can't be retransmitted.
So how long does it take for the transmission to happen? Is it pretty like just days or?
So the insect transmission is very quick. So it's a so-called non-circulative virus. It just binds to the mouth parts of the insect. The insect picks up the virus, and then the virus releases when the insect feeds on the next plant. And then it takes about 2 weeks for the virus to fully grow and infect the maize plant. One disadvantage to this system, of course, still is that we're dealing with viruses that are infecting maize plants. And this is because these are viruses that we know about. Sugarcane mosaic virus and foxtail mosaic virus are known viruses that we know about because they cause disease in maize and other agricultural crops. And so in the long term, these are not the ideal vector for the kinds of experiments that we're doing, because we're protecting the plant against insects, but we're also causing a viral disease in these plants. However, this, I think, is just a reporting virus. In any organism, there are lots of viruses which are just present. They're not causing disease. And the same is also true for maize. And what we specifically decided to do is we went out and looked for maize viruses that are not damaging to the plant. We did this initially in Iowa, and we found very few viruses in the corn plants in Iowa cornfields. And we decided that this is because we're not looking in the right habitat, and because there's very little infestation on corn plants in cornfields in Iowa. And so instead, we decided to go virus prospecting in Mexico, which is the natural origin of maize. We expected to see more viruses in these plants. And we collected leaves not only from corn growing in Mexico, but also from teosinte, which is the wild ancestor of corn. So that's me in Mexico with some teosinte plants growing in a sort of a semi-natural habitat. And so we collected leaves from about 100 or 200 different corn plants in Mexico, and then sequenced all of these samples to find previously unknown viruses. And we literally found hundreds of different viruses in these corn leaves that we collected in Mexico. And so we aren't to this point yet, but our longer term goal is to now find a virus that infects maize, that we can clone genes into, but that doesn't cause a debilitating disease in maize. So thinking about it sort of from a human virus point of view, what we're looking for is not the smallpox virus, but rather the vaccinia virus. So not the virus that causes massive disease, but the virus that we can use to cause an infection and cause protection against other things.
Great.
So our longer term goal, beyond the end of the current project, which is finished, is to take some of these viruses and look to see whether they replicate well in maize, but don't cause any really overt disease in these maize plants.
And so you mentioned the one project that you did against the fall armyworm. Did you also look at, like drought tolerant type genes, or is that something for the future, or?
Yeah, so as I mentioned, there were 6 labs involved in this project, and Claire Castile, who's now a faculty member here at Cornell University, she was trying to clone drought tolerance genes in maize viruses using the same 2 viruses, sugarcane mosaic virus and foxtail mosaic virus, and found that when she overexpressed natural plant genes that are turned on in response to drought, so basically expressing drought tolerance genes before the drought arrives, and she got plants that were then more drought tolerant because they were prepared for drying out. And so they recovered more quickly after they were hit by a drought event, again, in the greenhouse, not in the real world at this point.
That makes sense.
And so those were our 2 main targets. And on top of that, also the other labs are involved in the virus engineering part, to make improved viruses that we can clone lots of genes into and use them as vectors to change the physiology of the maize plant in sort of a temporary manner. So these are not viruses that are entering the genome of the maize. We're only altering the current generation of the plants. The seeds from these plants would be ordinary non-transgenic maize plants again. So sort of temporary transgenic maize to respond to a specific unknown threat.
Yeah.
And this fits with the longer term goals of DARPA that if, for instance, the corn is growing in the Midwest and some new insect pest comes over from Europe or Asia that we're not prepared for yet, we could quickly design a virus that causes resistance to that insect, release it in the cornfield, and save the corn crop. So as I said, DARPA tries to work on projects that are on the edge of what's possible. And this is certainly not possible right now, but we're developing the sort of toolkit to have a rapid response toolkit to a new threat that could hit the American food supply.
Are there any other practical applications for this type of technology?
Yeah, I mean, certainly people are thinking about this. And as far as I know, there's actually only one field trial that's going on, and that's related to citrus greening. And there's a company, Southern Garden Citrus, that has developed citrus tristeza virus as a way to combat citrus greening. Citrus greening is illustrated here on the left. This is a disease that's totally wiping out the Florida citrus crop. And at this point, after 20 some years of this, we still haven't developed a way to get resistance in the Florida citrus. And this is one of those cases where maybe we need to think more creatively and try things that are on the edge of what's possible. And so what they've been doing, if they've cloned an antibacterial protein from spinach into the citrus tristeza virus, infect the citrus trees here at Southern Garden Citrus, and are killing these bacteria that are causing the citrus greening disease. And that's showing promise. They've gotten approval from the USDA to actually do field trials, not just testing it in greenhouses and laboratory conditions. And that may be one way to combat this disease. One thing that they're doing differently from what we were doing in our DARPA project is that this is not an insect transmitted virus. So it's going to be a much more contained infection. You're only going to see infection in the trees that you're actually infecting with this genetically engineered citrus tristeza virus. And so this is, as far as I know, the only current approved real world application of a genetically engineered virus to rescue a disease in a plant or any sort of phenotype in a plant. And I think this is approved because they didn't come up with any other better solutions after 20 years of research for combating citrus greening in Florida.
And it really is incredibly devastating disease for those who haven't heard of it. It's wrecking all the industry down in Florida, and I think it's spread to California even as of a couple of years ago. So it's spreading up there as well. Are there any other examples you can?
As far as I know, no real world examples, but you can probably think of many ideas of diseases or abiotic threats that might be targeting your crop. I'd like to give one example that I've been sort of thinking about, and that's the illustrated here is the American chestnut. Historically, until about the 1920s, chestnut was an important tree here in the eastern United States. Probably 20% of all trees in the eastern US were American chestnut trees. Then the chestnut blight came along and these huge trees essentially all died out. However, their root systems are still alive, and all over along the eastern seaboard, we have these chestnut seedlings, or not seedlings, but sprouts coming up. They get to about maybe 6 or 10 feet tall, and then the blight wipes them out. So it affects the above ground tissue of these trees, but not the root system. So there's a lab at SUNY EFS in Syracuse, the lab of Bill Powell. They've now made transgenic chestnut trees that are resistant to the blight, and they're hoping to now repopulate the United States with blight-resistant chestnuts. But this is one resistant chestnut event. And in the meantime, we still have these millions of chestnut seedlings that are coming up every year in the forests, including around here in Ithaca. And so one idea might be if we could get a chestnut-specific virus and get a blight resistance gene and clone it into that virus, we could infect chestnut trees out in nature and thereby make them blight resistant, and thereby rescue not only a single transgenic tree with a gene that's been cloned into, but more broadly rescue the chestnut diversity that we have in the forest around here on the East Coast by making the existing trees blight resistant by infecting them with hopefully a benign virus that carries a disease resistance gene for the chestnut blight. But again, this would be a very far-fetched sort of idea. I don't think anybody's working on this, but it's fun to think of sort of ideas, what we could do if we had this sort of technology. And that again is the mission of DARPA, is to think of technologies that might be useful in the future and develop them before we even need these technologies.
Yeah, those are trees. That would be great if they came back again. So just getting back to the, I think one obvious question when I hear this, it's really cool science, but one of the obvious questions that pops in my head is, is there any danger here? I mean, could this technology be used? Could it be weaponized? Not just agriculture be weaponized, but could these viruses and insects be used as a weapon?
Yes, I mean, I think that certainly is something that people are concerned about, and there have been articles saying this, because the way this call from DARPA was proposed is, this is a defensive sort of thing. We want to protect our crops from, alien insects that might be invading, whether it's, something like the Colorado potato beetle or things we don't know about yet. But I could also imagine that you could engineer a virus that kills the corn plants, that's insect transmitted, that you might want to release in some other country, sort of a high-tech version of the Colorado potato beetle release. I think, yes, that's a real fear. I think the technology probably isn't there yet. And to be honest, it would be much faster and easier to do that with existing insects. So for instance, I mentioned this example of the fall armyworm, which probably was accidentally released in Africa. But if you wanted to damage somebody's agricultural economy, it would be very easy to take an insect that isn't there yet and release it and cause hundreds of millions of dollars worth of damage. And so I think probably at this point, it's theoretically possible to engineer a virus to damage your enemy's crop, but probably on a practical level, it's not possible. And there are many cheaper alternatives if you're a military person thinking along those lines.
Yeah, that makes sense to me. It'd be way easier and more, less expensive to do it the old-fashioned way, I guess. Okay, well, thank you, George. I think that's all the questions I have for you. Now it's time to take some questions from the chat box. If anybody has some questions, it looks like we have a couple in there already. What impact do you think this technology could have on humans?
Probably no direct impact. These are viruses that are, not infecting animals. you could have indirect impact. So again, a concern with any virus you release into the environment is you say, well, we're only going to infect the corn plants that we want to target, but what if the virus recombines with some other virus in the corn plant and then gets into native plants and then wipes out insects that we might consider beneficial? So I think this is a technology that would be very, very difficult to control simply because we don't know enough about it. And because, as I mentioned, there's so many other viruses in the corn plant, we can't guarantee that the virus that we're putting in the corn plant, stays in the corn plant. And so I think there are severe environmental concerns from actually releasing any of these viruses.
Yeah, so maybe you need some like a virus that isn't normally in corn plants, so it's less likely to recombine with the.
That could be an option, but probably a virus that isn't normally in corn plants wouldn't replicate in corn plants.
Yeah, it wouldn't work anyway, right? Right.
So, I mean, I would say this is not a technology that you, at this point at least, want to use for routine application, but maybe have on the back burner for emergency use, like for instance, the citrus tristeza virus, where there really has been no better option so far.
Right. Here's a question about the BT corn. In addition to the GMO method to integrate BT genes to the crops, would it make sense to directly spray the BT bacteria to the foliar parts of the crops or drench the bacteria to the soil where the crops grow to promote plant growth and induce host plant defense against phytopathogens and pests? I think that's done already, isn't it?
Yeah, so I mean, BT sprays have been around long before transgenic crops. If you go to Agway today, you can find 3 or 4 different BT sprays that you can use for your home garden or for your indoor plants. And so that was one argument for the development of BT crops, is that the same bacterial toxin that was already approved for spraying onto plants was then approved for growing in the plants. And so for that reason, yes, we can use BT sprays. It's probably more expensive in the long run to have it sprayed rather than genetically engineered in the plant. So that's one reason that the major agricultural companies were going with transgenic crops. But whether BT specifically induces pathogen resistance, probably not. That was the other part of the question. But there are certainly other things that people are working on, sort of inducers of bacterial or fungal resistance in plants that you could spray on the plant or drench the soil. I mean, a good example is obviously Ascribe, the company founded here through Boyce Thompson Institute that's working on nematode small molecules that would serve that kind of purpose.
Yeah, that's a technology of not actually killing things, but really boosting the plant's immune system. That's a very cool technology. Does anybody else have any questions? I'm not seeing a lot in there. I was wondering about if you could talk more about your search for viruses in teosinte, and are you looking at any other like near relatives of maize?
No. So the question is, where were we looking for viruses? No, we really only looked in teosinte and maize growing in Mexico. And so basically I drove around central Mexico with a pair of scissors and some Ziploc bags and was collecting maize leaves. I asked the farmers who were around, say, can I snip a few leaves off your plants? And they always said yes, it's a fairly harmless activity. And then I had a USDA permit to bring the leaves back to the United States. And then my collaborator, Steve Witham's lab at Iowa State University, they took these leaves and they sequenced, did deep sequencing to look for viruses in these leaves from Mexico. And as I mentioned, they found lots of viruses that we didn't even know about, including an interesting one. So there's a virus called maize streak virus, which is considered a huge problem in Africa. And it was thought that this didn't exist in North America, and there are lots of ways they tried to keep it from reaching North America. But in some random cornfield in Mexico, we found a maize streak virus. So my guess is that this is a virus that's present in Mexico, but it happens to be sort of a more benign version of maize streak virus. So it doesn't cause the massive disease that it causes in Africa, but it's just sort of resident in corn in Mexico, and we didn't know about it because it doesn't cause disease. And this, this is the kind of virus that we're looking for, sort of a non-virulent virus that we could clone something into and cause increased resistance.
Get a comment about the BT sprays from Tony Shelton. BT sprays onto foliage don't work very well because the proteins break down in about 3 days, and one needs thorough coverage, which is impossible to obtain. BT plants overcome these problems. Yeah, and I would imagine having to spray all the time, that's also, throwing a lot of greenhouse gases up in the atmosphere as well, which is a problem. Okay, one other question from David Silberman. Is it practical to use microarray analysis to identify useful specific gene sequences that could be engineered into viruses? And a second question is, once useful resistance sequences are identified, could CRISPR technology be used to incorporate those sequences directly into the target plant?
So yes, I mean, probably not with microarrays, but certainly DNA sequencing, genome sequencing can be used to discover genes that could be used to kill insects. And that's effectively what we've done is, we've gone to the literature and say, these are organisms that kill insects. What's known about the genes or the proteins that are doing this? And yes, of course, we could engineer those genes into plants. CRISPR is one technology that could be done. To do this, we could, if you wanted to specifically modify plant genes to make them more insecticidal is one option, or to use CRISPR to cut in a specific site and then insert our gene from spider or scorpion or lectin, whatever we want to do to increase maize resistance. But there, I think we're just recapitulating the technology that we have with BT toxins, and that we would go through the approval process of making a transgenic plant. And that would, at this point, probably take close to 10 years between when I decide I want to put a gene in a plant, I optimize the transformation, I generate these plants, I bulk up the seeds, I get approval, I get these seeds to farmers, probably is a 10-year process. And what DARPA was interested in was the rapid response aspect of this. And that's why we were cloning genes into viruses rather than into plants, because we can do this cloning more quickly. And this is a way we can get these genes into existing plants in one growing season very rapidly and say, here's a new threat, let's do something about it right now, is their vision. So I don't say that we've accomplished this, but that's the longer term vision of DARPA, is rapid response.
Right, so to kind of change the phenotype of existing plants rather than creating a whole new one and planting it from seed. Mm-hmm. Yeah, and you mentioned an interesting point earlier about, like if you're doing drought tolerance, for example, that takes a lot of energy. The plants you tend to expend a lot of energy on expressing these other traits. So if you have it all the time, even when there is no drought, then you'll probably be decreasing yield. Mm-hmm. And it wouldn't be working that well in the other years. Did I understand that correctly?
Yes.
Okay, we have one more question. Is there any possibility for the engineered virus to get prone to mutation, like viruses used to evolve continuously extensively? And further, does lateral gene transfer may affect the efficacy of the engineered viruses?
Yeah, so that's a question from Hassan. And yes, I mean, that is a concern. Viruses are constantly evolving. I mentioned that we have to clone very small proteins into the viruses because viruses are sort of optimized for having small amounts of DNA. If we clone large genes in there, they tend to recombine within the virus and the virus loses our gene that we've put in there. So basically what happens is we're selecting for viruses that are losing our transgene because the virus that doesn't have that extra piece of DNA or RNA in there replicates more slowly, and we're selecting for viruses that lose our gene of interest. So that's a disadvantage to this system, is that we have to work very carefully to either have small proteins or maybe optimize the virus, take some other virus genes out of there before we put our gene in to keep the virus from getting too big. And the other concern that, that we have, which is something that's difficult to address, is that viruses recombine with one another. So if we have our genetically engineered virus in the plant and some other virus comes in and infects that plant and is similar enough to our virus that it can recombine, we could have our gene go into a wild virus that's not been engineered, so it's not insect transmissible, for instance, and then our genetically engineered virus would be escaping into the environment much more readily just because of natural recombination within the virus. And so that's a big concern, which I don't think we've addressed, and it's going to be very difficult to address because we don't know the sort of the unknown unknowns here. We don't know what other viruses might be infecting the plants that we have our viruses in there. And so for that reason, all the experiments that we did were done in under high containment. They weren't actually even done here at the Boyce-Thompson Institute. So when we were doing transmission experiments with genetically engineered viruses, they were done in a biosafety facility at UC Davis. So this is a highly contained facility. To go in there, you have to get undressed, take a shower, put on a Tyvek suit, and then you work with your plants, and then the same thing on the way out. And anything that goes into the facility can't be taken back out. So when we had a camera crew taking pictures in our virus transmission experiments, they had to leave their cameras in there for 2 weeks in their freezer to kill anything that was possibly on those cameras, and then they could get their cameras back out. And so all their pictures were transferred out electronically. So it's a super contained facility for doing these experiments, just to make sure that we don't get that sort of virus escape that I just mentioned.
So freezing, there must be pretty cold to kill a virus. I didn't realize.
Yeah, in that case, they weren't concerned about viruses. They were concerned more about insect pests. It
Gotcha.
wouldn't be the virus that they were concerned about, because that's a facility where they were working with all sorts of pests and pathogens that are not native to North America. So it's one of the few sites in North America where you can work with insects and pathogens that are just not found here, but you're studying, how can we develop plant resistance to them? And so that's why we did our genetically engineered viruses experiments in that facility.
So it sounds like this is still quite a ways away from prime time, so to speak. There's still lots of concerns and technical hurdles. Yeah,
so I think that's quite true. And A, it's a technology being developed, and B, it's sort of a back burner technology. If there's a real emergency, it could be something that could be activated when it's needed.
Yeah, it's very neat. Very clever idea. It's very cool. Are there any other questions out there? I don't see any more in the chat box. Maybe we'll give folks a little more time. Could you think of any other examples? We talked about citrus graining disease and chestnut trees. I'm wondering if there are any other potential uses for this you could think of off the top of your head. I mean, it seems like there's all kinds of introduced diseases and insects and things coming all the time. Yeah, I mean, there's long lists of insect pests that would be found in other countries that we don't want to have here. And there's also periodic introductions like the medfly in California. Maybe one way, there's a whole arsenal of tools that they use to combat medflies every time we have an outbreak of the Mediterranean fruit fly in California. There's insecticide sprays, but maybe, transgenic viruses, targeting medflies would be one part of that arsenal to control medflies coming in. There's a note here in the chat, elm trees would be a good target, Dutch elm disease. Can we make viruses that target Dutch elm disease? So the American elm has been wiped out by a pathogen, but we still have elms around. Maybe we can infect those with a virus that make them disease resistant to the Dutch elm disease. You could also think about emerald ash borers or other more recent introductions. The ash trees here in the eastern US are being completely wiped out by emerald ash borer.
because there's no natural resistance in there, but maybe we could put in a emerald ash borer resistance gene into the ashes that are still out there. Or, beech trees are being wiped out by diseases here in the US. Again, is there some way we could make a virus that provides resistance to diseases in beech trees? And so the list is pretty much endless in terms of the number of pests and diseases that we would want to look at. The question is just, at what threshold do we want to use a fairly risky technology, and how economically important is it that we do this?
Great. One I've been hearing about in recent years is it's like the spotted lanternfly or something like that. They just popped that in. The spotted lanternfly, which is I think is a really neat looking insect. It's kind of pretty, but it's devastating, I hear.
Yeah, I mean, it's actually been seen here in Ithaca for the first time last summer. And so it's probably just inevitable that we get the spotted lanternfly here. And it's clearly a problem on trees, and agriculturally, it's probably the biggest problem for grapevines. So there's some grape cultivation that's been completely wiped out in Pennsylvania. That's in the chat here where spotted lanternfly was originally introduced. And again, right now, we don't have any technology to really combat the spotted lanternfly that's been infesting the grapevines in Pennsylvania. And almost certainly will be hitting the grapevines here in New York State in the next few years, given that there are already insects found in Ithaca. It's probably too late already to keep the infestation from hitting us here.
That's too bad. It also makes me think about, in terms of crops, there's still people who are pretty anti-GMO. Like, are there some crops that this just wouldn't fly for market reasons? If people, would people want to drink wine from grapes modified with this? I don't know if there's a good answer to that, but
no. I mean, one thing they're looking at with grapes, not necessarily with spotted lanternfly, but with Pierce's disease, which is a disease in grapes, a bacterial disease that infects the xylem of grapes, is all grapes are grafted. And what if you made a genetically engineered rootstock and then grafted your, Cabernet Sauvignon or whatever high quality grape on top of it, so that the disease resistance would come from the genetically engineered rootstock and spread into the upper part of the plant. So the grapes that you're harvesting are not genetically engineered, but the rootstock is. And so that's sort of a technology that's probably out there, but hasn't been tested yet, either for regulatory approval or for consumer acceptance. Is a grape that's grafted onto a genetically engineered grape considered transgenic? That's not known yet, but it's something that's going to be hitting us in probably the next few years, because that's something that people are looking at for combating Pierce's disease in California.
That's a good idea. I think they look for the genes, so probably not. It probably would not be considered a GMO. So that's interesting. So,
but they're clearly, let's say you clone a resistance gene into that rootstock, and it makes a small molecule that moves up into the grapes, or it makes RNA that's moving systemically in the plant, and then making a protein that's killing these Pierce's disease bacteria. It's sort of a gray area whether that would be considered genetically engineered or not. And as I said, it hasn't been tested yet, but it's certainly going to be tested very soon.
In fact, something similar could be done with a citrus greening disease, and a lot of those are grafted as well, right?
Yes, that's certainly the case, that you could consider making citrus that's grafted onto a rootstock that's genetically engineered and then passes resistance molecules into the upper part of the plant. I'm sure that's something that they're thinking about when they're doing research on citrus greening also.
Yeah, there's a lot of work on that. Okay, we're close to 1 o'clock. I think last call for questions. Is there anything else you'd like to add, George?
I don't think so. Just the summary is that this is, interesting but potentially dangerous technology. But I would say it's definitely back burner at this point, but potentially useful in certain situations.
Yeah, it's very, very neat stuff. Well, thank you again, George, for taking the time to chat with us today. And thank you everyone out there for joining us out there in Zoom land. And please join us for BTI's next Breaking Ground discussion series. Wednesday, March 30th, we will be here with Dr. Alexandra Skic, also known as Ola, who will be talking about her research searching for metabolites that could be valuable. And that's going to be a really good one. So please don't miss it. And you can go to btiscience.org for more information and to register. And I am going to put a bunch of URLs into the chat box, including the one I just said. So you can all have that and make it easy to register. And you could read more about BTI's current research and many other neat stories about BTI science in our annual report, which you can find online at btiscience.org/annual report. And BTI is an independent nonprofit research institute, and we operate in large part thanks to the generosity of community members like you. And if you'd like to make a gift to support BTI, donate online at btiscience.org/give or email our development team at development@btiscience.org. And thank you all out there. Hope you have a good rest of your day, and thank you for your interest and support of the Boyce Thompson Institute. Have a wonderful day and be well. And I hope to see you next month.